Can you imagine discovering an entire population of ancient humans from a single finger bone? Humans so close to us genetically that millions of people today have pieces of their DNA. DNA that affects how they breathe, how they fight diseases, even how they survive extreme environments. Today, we're learning about the Denisovans by looking at 10 things that we feel pretty confident we know about them, as well as their connection to the Dragon Man. We'll also ask a question that many people of faith are curious about. If Denisovans were real humans like us, where do they fit into the biblical story of Adam and Eve? Let's dig in. My name is Kathleen, and I have a master's in anthropology with an emphasis in archaeology. I'm also a Christian, and to be more pertinent to this conversation, an old earth creationist and probably what you'd call a theistic evolutionist. I like to lay my bias right out front. This podcast looks at things that I believe are really fascinating archaeological discoveries and sometimes how they align with the Bible, sometimes how they challenge it, and sometimes how they push us to think more deeply about what Scripture is and isn't trying to say. This is a place where you don't have to leave your faith or your brain at the door. Go ahead and click on that subscribe button so you don't miss future episodes. In this series of videos that I'm doing on the different hominins, the different of the Homo genus, what I'm trying to do is I want you to walk away with both a scientific understanding of that hominin and that species, as well as a faith understanding about how they might fit into that biblical picture. So the first part of this video is going to deal with that scientific look at the Denisovans, and then the second part is going to look at how the faith perspectives on how they might fit into the Adam and Eve story. And then at the very end, I'll sort of give you where I line up. That'll kind of happen a little bit through it as well. But at the end, I'll sort of wrap it up with just my own thoughts on how everything goes. Before we get started, I might have even already pronounced it two different ways. This word Denisovan. How do you pronounce it? You will hear Denisovan, Denisovan. Honestly, I've heard it all ways. It's like schedule and sedule, potato, potato. But according to the Leakeys that are famous archaeological, they made a lot of famous discoveries in the archaeological world. They say the correct version is Denisovans. I personally pronounce it in all those ways, depending upon the day, the hour, the mood. So forgive me. I don't think it really matters. So let's talk first about how the Denisovans were discovered. So they were found in Denisova Cave, which was located in the Altai Mountains of southern Siberia. It's a pretty cool story. We're going to dig a little deeper into that cave in a future video. But just a really fun fact is that the cave was named after a hermit named Denis, who was a devout Russian Orthodox Christian who lived in the cave in the 1700s. But starting somewhere around or before the 1970s. So it being called Denisovan Cave or Denisova Cave had nothing to do with the archaeological discoveries. It really just had to do with this hermit that lived there. Because it wasn't until maybe around the 1970s that there were a couple of Russian archaeologists that started excavating the cave. They were led primarily by Michael Shumkov and Anatoly Derevyanko. And these were of the Russian Academy of Sciences. So when in 2008, the archaeologists uncovered a small fragment of a finger bone, a distal phalanx, the cave was already known for Paleolithic artifacts and Neanderthal remains. In fact, hominins had been living off and on in the cave for about 300,000 years. So you think about this little hermit. I don't know if he was little. I don't know why I called him little. But this hermit that was living there must have been something attractive about the cave for him to live there. And obviously, it was also attractive to all of these hominins that were also living there for all of that time. And so 300,000 years, that's a really long time for these hominins to be going in and out of that cave. So when they found this bone, they assumed at first it was Neanderthal or a modern human child due to its small size and shape. No one suspected it represented a completely unknown human group. It was found in a layer. And these are the layers that they've mapped out, the different strata in the cave, and it was found in a layer dated to around 48 to 50,000 years ago. Now, you might be asking how they dated that. I'm not going to go into that here, but I do have a video on dating methods, and so you can take a look at that whenever you want to get some more information on that. But that sort of explains how they do this dating in these caves. But Svante Pääbo, a famous geneticist. I don't know what it is about him, but he's one of these guys that when I see his name come up, I get really excited because he is just really, really brilliant. And he was the person who first sequenced the Neanderthal genome. His team at the Max Planck Institute for Evolutionary Anthropology in Germany, they were able to extract mitochondrial DNA. We're going to talk about that in just a second. But this was a process they had already perfected for the Neanderthal genome studies. And that's really the thing he's really, really famous for, is he's the one who did that. So you may be asking, how could they have gotten any DNA after so long? Especially when you might have learned from some of my earlier videos how difficult and rare it is for anything to be preserved for that long. But the environment of the Denisovan cave was ideal, because it was consistently very cold and very dry. And these conditions helped slow down that degradation of the DNA. So I listened to a discussion with John Hawks and Chris Stringer. Both of these are leading scientists in this area. And I'll put the link actually of that particular video in the description. And John Hawks made the observation that because of these particular conditions of the Denisovan cave versus those in warmer climates, because in a little bit, we're going to kind of talk about where maybe some other bones have been found and where we believe the range of where Denisovans lived. But we don't have the same opportunity to study DNA from bones that are found in these warmer climates because of the conditions of that. And what this can cause is what we call a sample bias. And I discussed that. Again, I'm not going to go into it here. There's so much in this video already. I may end up splitting it into two. So if it says part 1 on the description, it means I split this into 2. I'm going to try to get it all into one though. But there's my bias video talks about sample bias. And so this is a very, very good example of sample bias, where these bones found in this cave were really, really well preserved to get that DNA, where sometimes we might still find a lot of fossils out in other environments, but they don't preserve that DNA as well. So we don't have that same opportunity to get the same information. But so what they immediately realized from the mitochondrial DNA, and what would get everyone excited, is that it was not Neanderthal, it was not Homo sapiens, and it belonged to a totally unknown human lineage. And this is one of the very few times in history where genetics discovered a new kind of human before fossils did. The team published their findings in Nature magazine in March of 2010. And then next, the team analyzed the nuclear DNA, which again, we'll discuss a little bit more in detail later. But after this testing, the bone would be cataloged as Denisova 3 and known to be from a young girl who lived about 41,000 years ago. Eventually, other Denisovan individuals, more Neanderthals and unknown hominins, were discovered in the cave. Okay, let's take a break. I want to explain the mitochondrial DNA that I talked about and the nuclear DNA that I talked about, because these are really important, and especially as we talk about other things and as we talk about other things in other videos, and as you guys go on and watch other videos, it's really, I think, very important to understand these two different types of DNA. So mitochondrial DNA. Mitochondrial DNA, which is mtDNA, is a small DNA molecule found in the mitochondria of a cell. The mitochondria are organelles that produce energy. It is double-stranded, So it's a double-stranded piece of DNA. It is circular. I see it more a little bit more oval, but it's a little circular, and it has, and this is important, 16.5 thousand base pairs. Okay, so mitochondrial DNA, it's double-stranded, it's circular, and has 16.5 thousand base pairs. And it is separate from the DNA in the cell's nucleus and is inherited exclusively from the mother. Mitochondrial DNA contains 37 genes. It's double-stranded, it's circular, 16.5 thousand base pairs, 37 genes, and those 37 genes are essential for mitochondrial function and is used in genetics for tracking maternal lineage. It's used a whole lot for that, and so it comes up a whole lot, okay? And so this maternal inheritance, along with a high mutation rate, makes mitochondrial DNA a valuable tool for tracing long-term maternal ancestry when studying human evolution, because it provides that direct line to trace maternal ancestry through generations. And so sometimes you guys might say, like, well, how did they figure that out? Or how did they figure that out? And hopefully this kind of helps you. I'm obviously not a geneticist. I get very excited about DNA. I find it extremely fascinating. I feel it's a little above my pay grade to fully understand. So I don't want to go into too much and then give you guys the wrong information, but I tried to make sure I had all my correct information for this, and that is mitochondrial DNA and the importance of it. And then you have nuclear DNA. Nuclear DNA is in DNA. So nuclear DNA is that genetic material that is primarily located in the cell nucleus and contains the majority of an organism's genetic information. It is double-stranded as well, and it has 21,000 genes that include the mitochondrial genes, and it has 3.2 billion base pairs that are those rungs on the DNA helix that make up the two nucleobases. So, nuclear DNA versus mitochondrial DNA. Nuclear DNA, 3.2 billion base pairs. Mitochondrial DNA, 16.5 thousand base pairs. Nuclear DNA, 21,000 coding genes. Mitochondrial DNA, 37 coding genes. There's a lot more information in that nuclear DNA than the mitochondrial DNA, but the mitochondrial DNA has benefits to it that we don't get from that nuclear DNA. So when I'm saying base pairs, just in case I didn't make this clear a second ago, I want to make sure I kind of come back to it. So when I'm saying base pairs, so if you look at the helix, if you look at the DNA helix, you see those little rungs. They look like rungs on a ladder. And so those are made up of two nucleobases, okay? And they kind of come together like this. You can see it. They made them different colors on there. And so those are really important. That's part of that coding that DNA has, okay? So nuclear DNA, so we talked about mitochondrial DNA, is only inherited from the mother. Nuclear DNA is inherited from both parents. It's much larger, as you can tell. It's much more complex. It can tell us about traits. It can tell us about relationships and admixture. And it's a lot more complex to analyze, as you can imagine. And so we'll talk more about how that nuclear DNA fits into the picture later, but I wanted you to sort of have that base understanding about it for now. Okay, so there were additional discoveries in that Denisova cave. And so after the finger bone, the researchers found teeth, these huge molars, far bigger than Neanderthal or modern human teeth. They had these thick, massive roots. They had these broad cusps. And the cusps were sort of like the little pillowy looking things on top of teeth. It had a robust crown, so that crown is kind of the top. And it was a completely different shape from Neanderthal and Homo sapiens teeth. So they also found a long bone fragment, and that's Denisova 11. And this one's going to be really important. This is from a teenage girl genetically matching the finger bones population. Now, we're not saying they came from the same person. We're saying it matched that same population. We're going to go into that a little bit later. It's pretty exciting. And then they found some other kind of assorted kind of fragments of bones. And so it wasn't enough for a reconstruction, but it was enough to show that they were very physically robust. So then there were some discoveries outside of Siberia. So one was in Tibet, up on this Tibetan plateau in the Baishia Karst cave. Then they found excuse me for this one, I'm probably going to butcher this one, but Shiaka jawbone, which was about 160,000 years old. It actually had 2 large molars still attached. Then through paleoproteomics, where they look at the ancient proteins, they were able to confirm the Denisovan identity. And this is a really exciting type of study. So we're going to do a little short break, a little short learning break here. Paleoproteomics is the study of ancient proteins from organisms that lived long ago. And it uses techniques like this soft ionization mass spectrometry to reconstruct the past life, the evolution, and the environments. And it extends this knowledge beyond the limits of ancient DNA, which is called aDNA analysis. It commonly uses dental calculus, which they did here, and because that dental calculus is a very common source for these proteins. The proteins can also be detected in ancient bones. The proteins can be found in almost all biological tissues. And so, like before plastics, they made up a large proportion of human-produced material culture. So it's a really, really useful thing to study and gain. So these proteins can be used to identify species and compare them over time. Proteins offer information that other archaeological or paleontological techniques can't access. They can even reveal what humans ate thousands of years ago. They can reconstruct ancient trade routes. They've been used for that. They can identify bone fragments. They can study diseases in ancient populations. So just so much information can be gained from these proteins. So an advantage over the aDNA, the ancient DNA, is that these proteins can remain intact for even millions of years, whereas DNA typically lasts just a matter of thousands. And so it's particularly in these protected environments like the bones and the teeth. So they also found a rib fragment, Shiaka 2. Shiaka. I think I'm putting the wrong emphasis on the wrong syllable, but it's also linked to the Denisovans. And there's possibly others, but we will discuss those later when we discuss Homo longi, aka the Dragon Man. You guys are gonna. I found it fascinating. I think you guys will too. But I mean, I just find the Denisovans so fascinating. The fact that, we believe they lived for this really, really long time, and yet we have so many or so few fossils. And then to have only know them through these genetics, or mostly know them through genetics. I mean, it's just such a crazy mystery, but also it's just so exciting that science has been able to achieve this and all the information they've been able to gain. That's just crazy. Anyway, it's a really neat mystery. I think it's going to be really exciting to see what the future brings. And we're also going to talk a little bit about why it is, and the 10 things that we know. You're going to find out a little bit more too about how we know that they were definitely around, okay? And the success they had when they were. So one thing that's interesting about the bones they found in Tibet. So Tibet has an average elevation of over 4,000 meters, which is about 13,000 feet, or greater than 13,000 feet. That's higher than Mount Everest. It's this plateau region with the largest space and highest sea level in the world. And this is huge because it shows the Denisovans lived at extremely high altitudes long before modern humans ever did. So now that we have a bit of a background on how they were discovered, let's talk about those 10 things that we think we know about them, okay? And as we go through these, there wasn't a great way to list these. So if one of them brings up a question for you that I don't immediately answer, stick with me because most likely it will be answered by another one. what I tried to do as I was researching these is that if one discovery brought a question to my mind, I really tried to answer that. But I'm sure I missed some of y'all's questions. And so I'm really looking forward to hearing y'all's comments on these and what other questions maybe I should have asked that maybe I can answer in a future podcast, or just answer in the comments and keep that discussion going. That would be really cool. Okay, so number one, what we've already talked about. They are known primarily from their DNA, not their skeletons. We only have a handful of bones. We have the finger bone. We have several molars. We have a few long bone fragments, including that long bone fragment, the Denisova 11, from the teenage girl found in Denisova cave. Then we have the partial jaw from Tibet. We have a handful of unidentified pieces. And then the morphology of some of these bones suggests that Denisovans had distinct physical traits, though we still lack a full skeleton to know exactly what they look like. But their genomes are beautifully preserved, giving us more insight into the Denisovans than into groups we have dozens of skeletons for. So that's just really cool. Okay, number two, they were a distinct group of archaic humans. So after sequencing that mitochondrial DNA that we talked about in about 2010 or 2012, Pabo's team then sequenced the nuclear genome, the nDNA, from the same finger bone, and that yielded remarkably high quality ancient DNA. And so while that mitochondrial DNA is very tiny and contains only a few genes, relatively few genes, nuclear DNA, like we talked about, is comparatively very huge and contains thousands of important genes. So the nuclear DNA showed that Denisovans and Neanderthals were sister lineages that diverged more than 390,000 years ago. And their common ancestor was likely Homo heidelbergensis. They split after the Neanderthal sapiens divergent about 600,000 years ago. Now, one of the things that I said before when I was saying about these, I called it 10 things that we think we mostly know about them. And this is one of those where they lie in this sort of common ancestor, heidelbergensis, where they split from Neanderthals. This is pretty sure what we know, what I just said, what I just talked about. But there is some questions about, like, if you look at their genetics, it says this. If you look at the fossils, they say this, and where they exactly fall on that there's just all questions all over this one. But this is what I think is the most accepted one, is that Denisovans and Neanderthals were sister lineages. They diverged more than 390,000 years ago. Their common ancestor was likely Homo heidelbergensis. They split after the Neanderthal sapiens divergence, which was about 600,000 years ago. And this makes them a unique branch of the human family tree. Then they lived mostly in Eastern Eurasia. And so you can see sort of here, there's a lot of questions about exactly where they lived, but we see where the Neanderthals were, and then we see this little area where they kind of overlap, and then we're up where Denisova cave is, up in Siberia, and then we see them all come all the way down, and but like the map says, that east-west realm isn't exactly known. Exactly how far east and west they went, but we will talk about that when we get to Homo longi as well, because that's where some of that guessing comes in. So one thing I want you to take notice of is if you look at the Neanderthals, so the Neanderthals go east-west, right? Like most of the thing is east-west, pretty much in that same kind of north-south area. So their climate was probably not super diverse, right? Like if you think about America, if you think about Maryland and Oklahoma, or Virginia and Oklahoma, probably about the same kind of weather, same basic kind of climate, not like Maine and Hawaii or Maine and Florida, So, but then look at the Denisovans. So they're going north south. So they're going through a very wide range of climates and of environments. So it's really, you just look at the fact that they were able to do that makes you kind of think, wow, they were able to really be adaptable. All right, number 3, they interbred with both Neanderthals and modern humans, so Homo sapiens. We see multiple gene flow events happening about 50 to 60,000 years ago. We see Denisovans and Neanderthals, we see Denisovans and modern Homo sapiens, and especially those in Oceania, Papua New Guinea, Australia, Melanesia, this whole area here. And so that's where we see a lot of this admixture coming. We also see them in parts of South and East Asia, where we're seeing a lot of that admixture. There were multiple, at least 2 to 3 genetically distinct different Denisovan populations that contributed different gene segments. And so you can kind of see from this little illustration, you see the 2 different Denisovan populations up there, and you see the Neanderthals and you see the modern Homo sapiens. And then if you kind of look on the map, it shows you how there was this different admixture in these different areas of the world. So the most famous example is that bone we talked about earlier, the Denisova 11. This was a long bone fragment discovered in 2012 in the Denisova cave. And the mitochondrial DNA revealed that her mother was a Neanderthal, her father was Denisovan, and that was the first generation hybrid ever discovered. And they nicknamed her Denny. That's why I wanted to make the comment earlier about the finger bone and this long bone, how I said, I pointed out that they came from the same population, but not the same individual. And when we're saying the same population, we mean that population of Denisovans, because there were 3 distinct Denisovan groups. So those 2 came from the same Denisovan group. Okay. Radiocarbon dating showed it to be greater than 50,000 years old. Look at my dating methods video. It was a 13-year-old, and they figured that out based on the cortical thickness of the bone. And so that cortical is that compact, dense outer layer on a bone. And that thickness changes with age, with sex, with genetics, hormones, and the location on the bone. So that's how they're able to tell things like that. That's one of the ways they're able to tell things like that. I hope you guys appreciate that. What I try to do is when I read about something they've discovered, like the sex or like the age, I'm trying to give you guys the tools to equip yourselves to go, it's not just a simple guess that they just so, oh, well, I think it's about this, or I think it's about that, or whatever. Like there's a lot of scientific methods that go into it. Now you can have questions about those you can have questions about it. You can do you. What I mean by that is that you can certainly question the science of it, but I think it would be disingenuous to treat it as if they're just guessing, because they are looking at patterns. They're looking at scientific methods of dating. I'm trying to equip you guys with some of the information of how they come to these conclusions. So a better method may come along someday. Science is always trying to improve on itself. It's always trying to come up with a better method to get a clearer picture, to get that better answer. If that changes in the future, if these answers change in the future, it doesn't mean that they're lying now. It means that they are using the best methods they have now to come up with the best answers they know now. And so using that cortical, and that's something we can see now on people, is that cortical thickness of the bone. And so it makes sense that they could use it on this bone as well and figure out the age, because we see that now. Could there be changes to that? Could there be, because I think one thing that we know is that I believe, I know I've read this. This isn't something that I've really focused on a whole lot yet. But I believe Neanderthals developed quicker than Homo sapiens. So is there a little bit of that? And so could we say that maybe it was around a 13-year-old? But I think what we know is it wasn't like a 50-year-old. And it wasn't like a 2-year-old. So I think just to kind of explain that could they be off, by some years here or there? They could for sure. But I still think it's a pretty good ballpark figure. Number 4, some modern populations carry significant Denisovan DNA. We've talked before about the Neanderthal DNA, and I think probably a lot of you guys have heard of that, where people today carry Neanderthal DNA. Do you know that there are people that also carry Denisovan DNA? Now, what's interesting, it's not as widespread as the Neanderthal DNA, and there's some more interesting things about it. So some populations today in Oceania have as high as 4 to 6% Denisovan DNA. So varying sources did have some slightly different percentages, but overall, a study in 2021 revealed the highest percentage is found in the ancestral Philippine Negritos. This is a small indigenous group surviving on the Bataan Peninsula on the Philippine island of Luzon, known as the Aita Magbukan. Hopefully I got that right. The Aita Magbukan is 5 to 6%. Then the Papuans and Melanesians and Australian Aboriginals are 4 to 5%. And then you have the East and Southeast Asia, 0.1 to 0.5%. And this includes China, Japan, and Korea. And East Asians harbor ancestry from at least 2 distinct Denisovan populations. And then here's interesting, Native American populations also carry around 0.2%. And that's something for, I am going to do a video on the first American. So I have a feeling this is going to come up again in that, although I know that video is going to be a little ways away from now. But I think we already know that Native Americans originated from Asia. So that's really interesting. And this is going to come into play again in one of the next things we know. Well, I keep having technical problems with this video. And so I am back again, future me, trying to re-record the last part of this. Actually, not the last part of this, more than like the last 3/4 of it. I don't know if I just need to buy myself a new microphone or what's going on, but I couldn't figure out a way to fix it with the software, so I'm having to re-record. Anyway, so number 5, Denisovan DNA affects modern human traits. And this is where things get really cool. So first, teeth. Now, those 3-rooted teeth that we talked about before, that is very, very rare in non-Asians. It's less than 3.5% occurring in non-Asians. But in contrast, its presence in Asian-derived populations can exceed 40% in China and the New World. And its presence in a 160,000-year-old archaic human in Asia strongly suggests that that trait was transferred to Homo sapiens in the region through interbreeding with archaic humans in Asia. So another way that their DNA affects modern humans today is through this EPAS1 gene, which has to do with high altitude adaptation. It is the EPAS1 stands for endothelial PAS domain protein 1, or called the EPAS1 gene. That's why we call it that instead of that long word. But it helps regulate the body's response to low oxygen levels by producing more hemoglobin. And that's that protein in our red blood cells that transports oxygen around the body. This is found in Tibetans. Besides finding that jaw on the Tibetan plateau, this also gives us an idea that the Denisovans probably lived in high altitude environments and had this physiological advantage in those high altitudes of the Tibetan plateau. This EPAS1 gene is allowing these Tibetans to survive in this extremely low oxygen atmosphere. Another way that Denisovan DNA is affecting modern humans is through immune system adaptations. There are some genes that aid in fighting infections that appear to have Denisovan origins. One is the HLA-C gene variant, and the HLA is a human leukocyte antigen. And HLA proteins are found on most cells in the human body. These are crucial for recognizing and fighting pathogens, strengthening modern immunity. And so it's believed this particular variant, the HLA-C variant, came from interbreeding with Denisovans because studies found that specific HLA-C alleles in modern humans. And HLA-C alleles are strongly linked to Denisovan DNA. So it suggests that this is where they were inherited from. There is also the MUC19, and this is a mucus barrier gene. And research study done at the University of Colorado Boulder focused on this gene. And what this does is it codes for mucin proteins involved in producing mucus. I know it's kind of gross. But the proteins will protect tissues from pathogens and also play a role in immunity. A genetic analysis of ancient remains and modern populations looked at this specific Denisovan MUC19 variant. And what it does is it shows this very specific, longer MUC19 sequence. It had a unique structure with extra repeats on it. And this likely helped early humans crossing into new environments, even like the Americas, fight new viruses and bacteria. Now, there was this Oreo like gene sandwich of archaic DNA. And this gene sandwich, this Oreo gene sandwich, points to Denisovan and Neanderthal interbreeding. That's what we talked about before, that offers this pathogen defense. And this means that this was a hybrid DNA segment of Denisovan and Neanderthal DNA, and it would get passed down to modern humans when they interbred. This process created a situation where Denisovan DNA sits between Neanderthal sequences, which is why they call it a gene sandwich. And that University of Colorado Boulder study showed that the gene is highly frequent in indigenous Americas. And this increased in frequency over time, perhaps because it offered an evolutionary advantage. But why didn't this protect them from European diseases? Well, there's many reasons, but they boil down to the millennia of isolation from Eurasia. The variant was beneficial for local diseases only, but it became unfortunately harmful against the novel viruses and bacteria that was brought over. And so this shows a specific genetic mismatch with European pathogens. So in essence, indigenous Americans' immune systems were just differently adapted, making them unfortunately extremely vulnerable to those specific diseases that Europeans carried, which we know led to these devastating pandemics. So these immune-related genes might have played crucial roles in protecting ancient and modern humans from South and East Asia, the Americas, and Papua New Guinea against specific pathogens. Overall, having Denisovan genes potentially helps populations like Papuans and indigenous Americans survive new infections, though some variants also raise risk for autoimmune issues like lupus and psoriasis. And all of this illustrates how Denisovan heritage continues to affect human health today, which is just wild. Another way that their genes live on in modern humans is through cold adaptation and metabolism genes. Now, one is the SLC30A9, or Salute Carrier Family 30 Member 9. And this is a gene encoding a protein that acts as a mitochondrial zinc transporter. And this is crucial for maintaining zinc balance within cells and mitochondria. So basically, it regulates zinc levels. Mitochondria plays an important role in skeletal muscle energy metabolism and converts the nutrients into energy. A Denisovan variant of this cell alters intracellular zinc balance, and by doing that, it impacts cell metabolism and function, which provides a survival advantage in cold, harsh climates. But this is also known to be associated with severe neurological issues such as profound ataxia, which is like a lack of muscle control and coordination, also intellectual disability and developmental regression. And then we have the TBX15 and the WARS2 genes. Now, these two genes are crucial for body fat distribution, metabolic health, and skeletal development. And potentially, this affects the expression of brown fat. And brown fat is involved in the rapid production of heat in hibernating animals. and even human babies. By altering that fat storage, it actually benefits northern climate populations in Siberia, in the Americas, Asian, and the Arctic. For example, Inuit populations carry Denisovan genes that help to regulate body fat and maintain warmth. So this kind of gives them some enhanced physical abilities. So modern humans who carry Denisovan DNA, we also see having increased muscle strength and endurance. All right, number 6. Denisovans likely had several genetically distinct populations. We've mentioned that before. We're going to kind of go into it a little bit more now. So researchers analyze this DNA from the ancient Denisovan remains, and then what they do is they compare it to modern human genomes. And what they did is they found different Denisovan variations in different populations. So genomic analysis shows at least 3 distinct populations, and that they seem to have split apart somewhere between 363 to 283,000 years ago. And they designate these, the geneticists designate these as D0, D1, and D2. So D0 is those Altai Siberian Denisovans. These are the classic Denisovans, the ones that are represented by the fossils from Denisova Cave. Their DNA is found in East Asians and some South Asians, but in smaller amounts than other groups. Then you have D1, and this is largely restricted to those Papuans and other oceanic populations that do include the Aboriginal Australians. So some scientists believe that they became part of the modern human genome maybe about 15 to 30,000 years ago, so not that long ago. But they are a more divergent group. This lineage contributed to traits like enhanced immune responses and even brain development. And then you have D2, the Eastern Asian and Papuan Denisovans. And this is a wider geographical distribution that spans most of Asia and Oceania. It left its mark on East Asian populations like Han, Chinese, Japanese, and even some indigenous Americans. So the D2 is the most distant from those D0 Siberian Denisovans. And it is actually so distant that some scientists do suggest that this group might need its own name, I guess, other than D2. So Denisovan is kind of more like a category than a single unified group. And there is some level of debate if the population should be their own species, but we'll talk more about that later. So modern humans encountered and mixed with these distinct Denisovan groups as they migrated across Asia into the Pacific. So we're going to look at this diagram again, this illustration again. So this illustration only puts 2 groups of Denisovans on here, but we have the modern humans, we have the 2 groups of the Denisovans, we have Neanderthals, because I still wanted you to see this one because I wanted you to get an idea of how they were spread across the world and how this admixture was happening. And so you see that there's these 2 waves of Denisovan admixture detected in these East Asians. And so you see over here by Australia, you can sort of see you've got the black with those modern humans mixed with one of the groups of the Denisovans and then the Neanderthals. And then if you look up here more like East China, you can see that it has all 3. It has the Homo sapiens, it has both groups of those Denisovans, and then you have the Neanderthals. And so that's why we're seeing these 2 waves of Denisovan admixture in these East Asians. So their physical appearance is still mostly unknown due to that low number of fossils. So that's another thing that we kind of know about it. But that one is getting clarified more and more. I think it's not going to be too long before we know a lot more about that. But right now it's still flipped up in the air a little bit. But there is a lot that we do know, and a lot that we know. There's a lot we know that they look like, and a lot we know that they did not look like, because of these genes and because of some of the fossils that we found. But because that physical appearance is difficult to determine, you'll notice I don't use a lot of reconstructions. While I love reconstructions because I think they help us get a good image in our heads of these different hominin groups, As particularly with the Denisovans, I find the information on what they look like is so sketchy that one thing that we do seem to know pretty good about them is that they were fairly robust individuals. But for some reason, though, I look at some reconstructions and they don't seem to be very robust looking in the reconstruction. So it's given me some skepticism on some of those reconstructions. I still love the art of it, and I still think it's a very, very useful tool, but sometimes I think it can give us the wrong picture. So I just felt like those were all over the place, so I'm not going to use them. Anyway, there's a long way to tell you that. Okay, so from the fossils, we can tell that they had these large jaws. We actually have a jawbone. We had these large molars. So you can see in this, you can see how small the Neanderthal and the present-day humans' teeth are compared to these very large Denisovan molars. And so also, like we talked about before, the molars were morphologically distinct. They had the 3 roots. The Neanderthal and sapiens only had or have 2. So you can look here. You can see how that tooth looked, that molar with its 3 roots looked. Pretty crazy looking. And the genetic analysis and anatomical reconstructions that were done based on DNA methylation, which is the chemical changes, and looking at those patterns suggest they had a wide pelvis, they had this elongated face, they had large joints, they had a large rib cage, and so most likely they shared sort of this robust build that was similar to the Neanderthals. So we still lack a full skull or skeleton to reconstruct them accurately. Or do we? No, I keep teasing that, right? Right? You just got to stick with me a little bit more. But hopefully you're really enjoying this information. I know I really enjoyed researching it. There was so much. And I didn't expect that because these are these mysterious Denisovans. But I think maybe that's what made the research even more. There wasn't much, but there was also a lot. The other thing is that they lived across a huge range. We sort of already talked about that before. There's the archaeological and genetic evidence places them in Siberia, the Denisovan cave, Mongolia, northern China, the Tibetan plateau, Southeast Asia, and these genetic signals suggest influence on the earliest Australians. Their range may have been larger than the Neanderthals. It's just hard to tell right now. They had this high altitude habitation long before modern humans were able to reach the 14,000 feet elevations. They were not primitive. They were capable, they were adaptable, and they were intelligent. Number 9, they may have had advanced cultural abilities. Material culture linked to them includes microblade stone tools. Now, in my video about the genus Homo, we talked about that importance of the stone tools. I've actually probably talked about this in numerous videos. The stone tools are such an important marker in the archaeological record because of what they tell us about the cognitive abilities of these hominins. And so they found these microblade stone tools. So the microblades are like teeny tiny ones, and they can be used for a number of things. Also ornaments and polished artifacts, including this green chlorite bracelet, which was a stone bracelet unearthed in Denisova Cave in 2008. I mean, look at that. That's gorgeous. Like, that's something we would find in a store today and want to buy. My favorite color is green, so I would absolutely be all over that thing. And so how they were able to date that bracelet was that the soil around the 2 fragments of the jewelry piece was dated with oxygen isotopic analysis to about 40,000 years ago. And it was also in the same layer where we found a Denisovan bone. It's fashioned from this imported chlorite. Now that's pretty important. Both the chlorite is fragile, but also it would have needed to have been imported from over 150 miles away because it wasn't native to that area. And so that's really interesting. So they were taking it, obviously recognizing that it was beautiful and traveling with it. And it was fragile. So they would have had to have taken really good care of it too. And they used techniques that were previously not considered characteristic for the Paleolithic era. They did this easel speed drilling. They had a boring tool type rasp. they would do this grinding and polishing with leather and skins of just this varying degrees of tanning. So there was wear near a hole that was drilled on the outer surface of the bracelet that suggests that it may have held this leather strap attached to a heavy charm. And that's pretty cool. Also, this sewing needle was found in the Denisovan cave in that layer ascribed to the Denisovans, and that was dated to about 50,000 years ago. So we're not really sure when hominins started wearing clothes, but we're pretty sure they were at this point. But this suggests, having this needle suggests the Denisovans possessed sophisticated technology and complex behaviors. And those complex behaviors suggest complex thinking. And so they likely, having those needles, they probably crafted their clothes, maybe even created tents using the needle. The first form of sewing was probably like tying together animal skins. So just tying them together. And then they might have used thorns and sharpened rocks as like needles. And thread was likely animal sinew or plant material. And so sinew is some of you guys probably know this, because I do think that that maybe if you're into hunting or something like this, you're probably a little bit more familiar with this. But so sinew is like that fibrous. I don't know for sure if it's the fiber pulled off of a tendon, but we know where the bone connects to bone. And so they would use the fibers of it and use that as their thread. I watch a show called Alone, and they always talk about the importance of using the animals. I mean, and you hear about Native Americans, you hear a lot of people talk about when they do go hunt, the importance of using as much of the animal as possible. So that they were using it this way. And then again, going back to that cognition, who looked at that and said, I can use that to connect this piece of fabric to this piece of fabric. And then I can put a hole in this piece of bone and sharpen one end of it to poke through to use as a needle. I mean, it's so second nature to all of us, but you think about that first person, that first hominin to come up with that process and to come up with that idea of doing that is, to me, it's just awe-inspiring when I think of that. So, but this probably this early method of sewing. So to get back, sorry. So probably this early method of sewing where they used the thorns and sharpened rocks as needles. It was limited. You can imagine it was limited because they didn't appear to have that ability to produce a small enough hole in a needle, such as a bone sliver, and not damage that material. And so they needed the technology to create that tiny hole. And the Denisovans were able to achieve this. It showed a technological advancement, possibly even more so than their Homo sapiens and Neanderthal contemporaries. But I'm never going to put down my Neanderthals. I love my Neanderthals. But it is really fascinating. I mean, it really does. I don't want to put down my Neanderthals because they were very smart too. And they had some really, really amazing technologies too. And obviously Homo sapiens, we've we're the ones that are still existing, and nobody's exactly sure why, but out of all the hominins that were living all at the same time, we're the ones still here. So we obviously had some pretty good things going on ourselves too, and we'll talk about that in a future episode. But this is really, really fascinating to me, as how they came up with that idea and were able to get that tiny hole in that bone fragment and use that sinew. Okay, I feel like I've beat that one to death. It's pretty cool. Okay, number 10. They coexisted with Neanderthals for tens of thousands of years. The Denisova cave alone shows that multiple generations of Neanderthals and Denisovans living in the cave. Some in separate layers, but some within the same layers. So the most famous example that we've already talked about is Denny, and just sort of to run through that again, that's Denisova 11. That's that bone fragment that was found. And that DNA and dating analysis showed that about a teenage girl, probably maybe around 13 years old, about 90,000 years ago. And what this also did, it challenged earlier ideas and notions that they were isolated groups. It provided crucial evidence for complex early human interactions. And as we come to find out, as we study more and more and we find more and more, these guys were interbreeding all over the place, and that's part of what makes it very difficult to put them into these very strict categories. Within the same vein about how much they live together, there are also multiple genomic studies that show Neanderthals carry Denisovan DNA. Remember that Oreo, that gene sandwich that we discussed earlier of the Denisovan and Neanderthal genes, and the Denisovan in the middle, the Neanderthals on the sides, that was passed on to modern humans. And so that's proof of multiple interbreedings. And then we also can identify the specific haplotypes and alleles to understand the archaic admixture of DNA. We're going to take another little learning break here because you just heard me use the terms haplotypes and alleles. When I studied DNA in school, I really found haplotypes were really fascinating. I'm not a geneticist, as I've talked about before, so I'm not going to go too far into it. I really want you guys to understand some of these terms, sort of just like what I said before. I want you guys to have a full understanding of what's going on here, so that you're not just hearing these terms. But if I can take these little breaks every once in a while, what I'm hoping to do is give you guys a place to put your feet when you hear about them, okay? So an allele is another name for a DNA variant of a specific gene. So you inherit one allele per gene from each parent, and the genes determine traits such as your hair color. The alleles are the code for the specific option of that trait. So like your gene determines your hair color, the alleles are going to code for whether your hair color is brown, blonde, red, etc. So that's what the alleles are. And the definition of a haplotype refers to a set of alleles, those DNA variants along a single chromosome, that tend to be inherited together as a group. So they tend to be inherited together because they are close to each other on the chromosome. So, for instance, this little illustration, so you have 2 copies of each chromosome. So one chromosome comes from your mother, one chromosome comes from your father, and each of these chromosomes carries its own haplotype. Mom passes one of her haplotypes, dad passes one of his haplotypes, and then the child receives one haplotype from each parent. And together, these form the child's genetic makeup for that region of the DNA. So that recombination, though, can mix things up. And this is where it's important. So sometimes chromosomes swap segments before being passed down, and that's called recombination. So large sections of a haplotype often stay intact, but smaller pieces can be mixed with the other chromosome. So siblings share haplotypes, but they aren't genetically identical. Haplotypes can be traced across generations, but they do slowly change over time as that recombination happens. When you see similarities in certain populations, what you're seeing is haplotypes, okay? So haplotypes can reveal historical admixture events. And so this is what those geneticists are studying. It can reveal these historical admixture events between modern humans and archaic hominins, such as Neanderthals and Denisovans, by showing these shared genetic variations. Haplotypes can be and are used to understand population bottlenecks, migrations, expansions, etc., all those kind of things that are going on with these populations. They are also used to provide insights into the inheritance patterns of traits and diseases. It's also how ancestry is determined by those companies like 23andMe, Ancestry DNA, and those types of groups. They are looking at haplotypes. So looking at the areas where they lived, it doesn't overlap very much, but where they did, that genetic DNA indicates that they interbred significantly. This paints a picture of a complex, interconnected, ancient world, not isolated groups that are living independently and separately. Okay, you're going to see a lot of iterations of me in this video because I keep having problems with my audio in this. If anybody knows how to help me out with that, that'd be great. But I just keep getting this reverb back. And so this particular section too, like you might see another iteration of me later in the video, but right now, I needed to redo this section. So talking about what the scientists expected the Denisovans to look like, it's now time to talk about Dragon Man. So first, let's talk about how Dragon Man was discovered. Back in 1933, in Harbin, Heilongjiang province in northeastern China, there was a bridge worker. Now, right now, at that point in time, the Japanese had invaded northeast China, and so the bridge construction was under Japanese occupation. And a bridge worker found this skull, and it was really huge and nearly complete. And he reportedly hid the skull in a well to keep it from being confiscated because he obviously must have realized its significance. But that skull remained hidden for decades until the man was dying and he told his grandchildren about it. So in 2018, it was finally studied scientifically. And then that scientific study didn't come out until 2021. So this was not that long ago. And in 2021, it was formally described and they called it Homo longi, which means dragon riverman. The estimated age of that skull was about 146,000 years old, putting it in the Middle Pleistocene. So its unusual recovery does put some questions in the scientists' minds and all that sort of stuff. And those things get considered in this study. But despite the unusualness of it, it was remarkably well preserved. And in fact, it is one of the best preserved fossils. And now, according to the research done for this paper from G et al., there were multiple dating methods applied. And these different dating methods showed its age from anywhere from 138 to 309,000 years ago. But the most reliable one showed 146,000 years ago. And so I think that's important for you guys to recognize. For one, they applied multiple dating methods to it, and they didn't just choose one in particular. They chose the one that gave them the most reliable finding. Now, there are key physical features about the skull. There was this mosaic of archaic and modern features. For instance, it had this massive brow ridge, and that's an archaic feature. It also had a very large cranial capacity, about 1,420 cc's, which is within or above the modern human range, and it's comparable to Neanderthals. And that is a modern trait. Then it had this long, low skull that sort of sloped back. That's an archaic trait. And then it had these large square eye sockets, and it had this flat face. And the flat face is a modern trait. The flat and low cheekbones is another modern trait. It also had this very large dental arch, which is an archaic trait. And it also had these very big teeth, which is an archaic trait. And what's even more interesting, so it had one tooth that remained in it. And that tooth had 3 roots. And remember what we talked about, how rare it is for a tooth to have 3 roots. And this one had 3 roots. So what's interesting about this is that this mixture of archaic and modern features, it didn't fit neatly into Homo erectus or Neanderthals or Homo sapiens. This is why Homo longi was considered a new species. And so the research team argued that the square eye holes, the long and low brain case, and the lack of a ridge along the skull's midline distinguished it from other fossils. And it's the combination of the features, not just 1 or 2 of the features, that does distinguish it and makes it unique. Now, the skull is too derived to be Homo erectus, but it's too different from the Neanderthals to be another Neanderthal. Yet, it's also not anatomically modern human. So they propose that Homo longi is a sister lineage to Homo sapiens, closer to us than Neanderthals are. And that claim was controversial from the start. You might remember earlier I talked about that nuclear DNA testing showed that Denisovans and Neanderthals were sister lineages. And these guys are proposing that Homo longi is a sister lineage to Homo sapiens. So this was one area where it wasn't fitting in with the Denisovans. And this continues to be a very difficult thing to parse out. So lumpers and splitters are always going to argue about these classifications. So why Homo longi was not considered a new species by some people is that they believe that many of the defining characteristics had more to do with its size than as distinct features, which may be just a matter of variation in a species. And variety can be seen by age, by sex, by regional adaptations, etc. Now, Chris Stringer, a very famous anthropologist, noted the similarity of combination of features with Dolly Man, which is Homo daliensis, which is another Eastern Chinese skull. But that Dolly skull is smaller, it's older. It's actually dated to 250,000 years ago. And so why do Denisovans matter here? We've already seen some stuff that fits in with what we might have thought that Denisovans were going to be, and we've seen some stuff that fits in with what we didn't think they would be. And so why do they matter here? Because we know Denisovans are known mostly from their DNA, not their skulls. In fact, no complete Denisovan skull had been definitively identified. And as we've discussed, there are very few fossils that are actually connected to the Denisovans. The Harbin skull is rare. It's large, and it's an Asian Middle Pleistocene skull. And this is exactly what scientists did expect the Denisovans might look like. Now, back in 2021, there was no ancient DNA able to be extracted from the Harbin skull. So the fossil had been chemically treated, and that kind of destroyed all the DNA preservation of it. But recently, new analysis came out with some new methods in 2025. So very, very recent. They were able to successfully retrieve mitochondrial DNA and protein sequences from the dental calculus, the calcified plaque on that Harbin skull tooth. And this confirmed a direct link to the Denisovan lineage, the one that's known from the Siberian cave, not one of those other populations. Now, independent paleoproteomic analysis, you remember we talked about paleoproteomics earlier, and innovative ancient DNA experiments on the Harbin cranium were done on its dental calculus. And this genetic evidence provided very strong support for the Harbin individuals being Denisovan. Researchers also noticed that the Harbin skull shares traits with suspected Denisovan fossils, like the Jiahe mandible. I know I didn't pronounce that right, but I don't think I'm ever going to pronounce that one right. But like that mandible that was already confirmed Denisovan. It had the right dental proportions, it had a robust jaw, it had very large molars, and predictions of traits based on Denisovan genetics, especially that robust facial structure, the broad flat midline, and those thick cranial bones. Geographically, it also fit with the East Asian Denisovan range in Siberia and Tibet and Southeast Asia. Between 2022 and 2024, several independent teams reanalyzed the Harbin skull using broader data sets and different methods, and they had an emerging consensus. So these were independent analyses. They began to have a consensus on what they were finding. Homo longi is very likely a Denisovan, or it's extremely closely related to Denisovans. Now, some researchers argue that Homo longi should not be a separate species from Denisovan because they believe it likely represents the best known Denisovan skull so far. But others prefer calling that Harbin individual Denisovan without renaming the species. And this Harbin skull offers the first look at the likely full facial morphology of Denisovans. Now, what do scientists agree on now? They have a pretty strong agreement that Harbin is very likely Denisovan or Denisovan related, that Denisovans were far more widespread and diverse than once thought, that Denisovans were morphologically diverse, that the large, robust skulls like the Harbin fit what we expect Denisovans to look like. And it may represent the best example of a Denisovan so far. Now, what's still debated? Now, whether to call the Harbin skull Denisovan or a Denisovan-related Homo. Also, whether Denisovan should be a species or a population within a broader Homo group. Now, what's no longer widely accepted is that Homo longi is a sister species closer to modern humans than Neanderthals. That original claim has pretty much largely fallen out of favor, although there are some debates on different ways of looking at the DNA and the morphology and the phylogenetics, all that kind of stuff, how they kind of line up. So it's still a big mystery here. Now, why does this matter? What is the big picture? If Harbin is Denisovan, now prior to this, we didn't have a lot of information about the Denisovans. We were curious, who are these people? We have their DNA, but we has so few fossils. And this tells us that the Denisovans were not these sort of like one-off kind of shadowy cave dwellers, but they were actually these dominant humans in East Asia. They weren't some like primitive side branch of hominids. It also tells us that Denisovans had large brains, complex regional adaptability, and that they were successful in extreme climates, which we kind of already knew from their DNA, but this just confirms that even more. So now let's discuss, how do Denisovans fit in the story of Adam and Eve? Now, I want to share with you some Christian authors and speakers that I probably mostly align my belief system with. One is William Lane Craig. I really like him a lot. He's an analytic philosopher. His organization is called Reasonable Faith, and he has a book called In Quest of the Historical Atom. And then you have Joshua Swamidass, and he is a computational biologist. I don't think he has like an organization, but I think he has a podcast called Peaceful Science, and he has a book called The Genealogical Adam and Eve. And then there's John Walton, and John Walton is an Old Testament scholar. Interestingly, he's also son of Sam Walton, who's a founder of Walmart. And he has a book called The Lost World of Adam and Eve. And then there's Hugh Ross, and Hugh Ross is an astrophysicist, and his organization is called Reasons to Believe. Now, each one of these I really, really like a lot. You'll notice if you look into them, they don't all agree with each other, and I don't agree with any one of them fully. So they're really interesting to look at, because they've got some really neat ideas. So I'd really kind of encourage you guys to check them out if you're interested. So let's first talk about what is human, because that's what this, I think, kind of gets down to. Were Denisovans human in the sense of Adam and Eve being human and created in God's likeness? Were they part of God's creation of being in his likeness, or were they just some other creation of God outside of being created in God's likeness? And that's where we know they were created, right? Because we have their bones. Like, we have that they were here. We have that there's a lot of differences to them than us, but there's also a lot of similarities. So I have a video I put out that talks about what defines the genus Homo. And it goes into that idea of how scientists classify the genus Homo, and Homo means human. So it kind of goes into how scientists do, but it also goes into, and that's what I want to discuss here a little bit, is what do we think it means to be made in the image of God? And those are going to intermingle a bit, because some of those things are going to fall into both. But what we talk about scientifically, what mix of features scientifically defines the genus Homo, is we're going to see a larger brain size. We're going to see it being greater than about 600 cc's. And that Harbin skull was 1450 cc's. So it was much bigger. We're going to see, with the skull features, we're going to see a more globular skull feature, more of a round skull feature, but not fully. None of these are like, okay, you have to have every single one of these things to be considered into the genus Homo. But it is a preponderance of having more of them. That's going to put them into that. Or they're going to be this transitional kind of species or whatever. But so with the Harbin skull, we do see that more elongated. So that's when we were talking about a more archaic feature and a more modern feature. And so that skull is going to be a more archaic feature. We're also going to see, defining the genus Homo, is a less prognathic face. And so instead having an orthognathic face. And so the Harbin skull is orthognathic. It's got a more flat face on it. We're going to start to see smaller jaws and teeth. But the Denisovan teeth, we know, are very large, and that jaw is very large. We're going to see habitual or obligate bipedalism. By nature of the activities that we have proof for that they were doing, bipedalism can be inferred to the Denisovans. But we also see in that Harbin skull, we see the location of the foramen magnum. And that foramen magnum says bipedalism, because that spinal column is going to be coming down straight down, not going out the back. We're going to see complex tool use. There are stone tools found in connection with the Denisovans, like that needle, but also some other typical stone tools that we find with other hominins. We're going to see signs of advanced cognitive behaviors. We're going to see abstract thought. We're going to see them making jewelry, not out of necessity, but out of a desire to create something artistic or adorn themselves. Now, just as to be fair, these are potentially linked to other hominins in the area, but the other evidence suggests these very easily could have been linked to the Denisovans. We see them creating that needle, and this abstract thought is looking at that material and seeing how it can be manipulated and used for something useful. We see their ability to adapt to a large variety of climates. We talked about that long range that they live in. And so we're looking at them living in the high altitudes of the Tibetan Plateau, which is over 10,000 feet, to the lowlands of Siberia, and even potentially the subtropical Laos and islands in Southeast Asia. We also see them having complex hunting strategies. There is evidence from the Tibetan Plateau that suggests the butchering of a variety of animals. So let's talk about the faith part of that. So what does it mean to be created in the image of God? Well, we see in Genesis 1:26 when God said, "Let us make man in our image after our likeness, and let them have dominion over the fish of the sea and over the birds of the heavens and over the livestock and over all the earth and over every creeping thing that creeps on the earth." And in Genesis 2:7, we see, "Then the Lord God formed the man of dust from the ground and breathed into his nostrils the breath of life, and the man became a living creature." So whether you take Genesis metaphorically or literally, the Bible does separate out the creation of humans from the creation of other animals. Man is the only one that he says was created in his likeness. And Genesis does single out this breath of life that God breathed into man's nostrils in the creation story. But later, we do see in Job 12:9-10 and Psalms 104 that the Bible kind of indicates this breathing life into all living creatures. But where the difference seems to be is there is this much greater intimacy shared when God is breathing into the nostrils of man. And even when he's talking about, with all the other animals, it was he just created them. It's almost like from a distance he created them. But with man, he shapes him. He gets his, it's this imagery, whether it's metaphorical or literal. It's this imagery of him getting his hands in and shaping like clay out of the dust of the earth, creating man. So what is God's likeness? This is a big question. Is it physical? Is it cognitive? Is it spiritual? Is it something else? The concept in Latin is imago Dei, and this emphasizes reason, morality, love, and relational capacity. And this is about possessing unique attributes that mirror God's character, allowing us to relate to him and others, and that divine sort of calling to steward the earth. And so this concept of both being made in the image of God and then being formed by God's breath, it sets man apart from the rest of creation. But what does this mean? And how does it set humans apart from other creation? And how do we see this in the archaeological record? And so for the sake of discussion, I'm going to kind of go with what I sort of posed in my Neanderthal video when I asked kind of the same type of question about the Neanderthals. So if Denisovans interbred with Homo sapiens to the point that their genetics can be found in our DNA today, does that mean that they too were part of God's image-bearing creation? Or were they part of God's plan using this evolutionary process and this interbreeding that was happening to pass these useful genes onto modern Homo sapiens, which were maybe this was his final plan? Or does he have something that we can't even conceive of now? So kind of reflecting on some theological viewpoints, there are some Christian scholars that suggest Adam and Eve could have been a historical pair chosen by God from within a much larger human population. So this idea, which is sometimes called genealogical Adam and Eve model, means that Adam and Eve weren't the only humans alive, but they were called into this unique relationship with God. Now, others take a more symbolic view, sort of seeing Adam and Eve as a story that expresses humanity's universal reality, our calling, our failures, and our need for redemption. And in that sense, Denisovans and other ancient humans aren't excluded. They're actually part of that broader tapestry of creation that the biblical narrative is trying to explain. So what do we do with this? We have these fossils of these early hominins. And so these early hominins can show us that the human story is possibly far richer and older and more interconnected than many of us grew up imagining. And far from threatening faith, this can actually expand our wonder of what God has done. And if Adam and Eve stand as representatives of humanity before God, then perhaps their story is big enough to include not just modern humans, but these early hominins, which are our ancient cousins as well. And it's a reminder that science and faith aren't enemies. They're just two ways of exploring who we are, where we came from, and what it means to be human before God. I think one thing that's really important when we are looking at this is like not to get too caught up, sort of like before where I was talking about how the genus Homo, like this is how scientists define the genus Homo. Now, how scientists define the genus Homo can change because they're expanding, and as they come across new discoveries and things like this, like science, constantly is correcting itself. And that's why we see it kind of change. It's not that somebody's trying to lie to us or keep something from us. They're just changing as they have new discoveries. They're trying to figure it out in a very scientific way. Those of us who have faith, we can say, hey, it's really exciting to see the science, but my faith tells me God did it anyway, no matter how it is. But looking at the science, we can just enjoy this really neat way of seeing how God might have done it. And so we want to be careful that we don't have to get too caught up in these categories that are just humans trying to put things into a box. God is not confined by those boxes that we do. He is a God of order. He's not a God of chaos. That's why we want to do that. That's part of being made in his image, is us wanting to put things into order. And so, but we have to remember that they were put in order. These categories were put in order by man, not necessarily by God. That's why he doesn't talk about them in the Bible. So I don't want us to get too caught up and too argumentative about human categories. And also, we can present multiple Christian perspectives about this without forcing any kind of single conclusion. For instance, one view, it puts Adam and Eve as genealogical ancestors. And from this genealogical Adam perspective, Adam and Eve lived in the ancient Near East. They became the universal genealogical ancestors of all humans by this intermarriage over time. The Denisovans and Neanderthals and maybe other early hominins would become part of that family line. Their DNA would have flowed into Adam and Eve's descendants through this natural interbreeding. And this means that there's not a contradiction with Scripture, because Genesis focuses on the theological lineage, not population genetics. Another view is that Adam and Eve were among a broader population. So this is an old earth evolutionary view where God creates Adam and Eve as covenantal representatives. And they lived in a world already filled with humans. Denisovans would be part of that broader population, and this interbreeding is expected as part of this view. And then there's another view of Adam and Eve as archetypal, not genetic. And this is a literary theological view that has Adam and Eve representing humanity. And then Denisovans are just simply another branch of early humans. And in this view, Scripture is not attempting to describe population level origins, All of these views allow Denisovans to fit into a faithful Christian understanding. Now, one thing that's kind of interesting that I read about, and this is just a little fun fact thrown out there, is I guess there's a lot of people who believe that Denisovans were giants. And I know the Bible does talk about giants out there, but I kind of want to press this issue. We have enough fossils from the Denisovans to know they were not giants. They were probably broad and stocky like Neanderthals, but Neanderthals were actually shorter than Homo sapiens. And there's a good reason to believe that Denisovans had a very similar body type and height as Neanderthals. So that myth that they were giants doesn't really have any scientific backing to it. So just kind of throwing a little fun fact in there. All right. So that was a little fun fact. And if you enjoyed this video, I just ask you to subscribe, like it, share it. I'm really excited to have all the subscribers I have. I thought I would have like 7, but if you could share or like it or subscribe to it if you haven't already, that is just a really that's just a free and very easy way for you to help out my channel, to get to other people, and to help me out to continue putting out more. So I hope you'll do that. And keep in mind something I'm doing. If you stuck with me this long, you're probably one of my deep dive people. But I have started to put out another version of I've started with this video because it's taken me so darn long to do. I'm going to start creating a shorter version, like a 15-minute version of these that I'm calling core concepts. So there's going to be a core concepts version of each topic and a deep dive version of each topic. And the core concepts is just going to condense everything into just the essentials. So if it's a topic you're sort of cursory kind of interested in, you're sort of kind of interested in, that 15-minute one might be for you. Or if you are like, no, that's something I want to know more about, then, the deep dive's for you. I hope you guys will keep supporting me and I hope you're enjoying this. Please let me know in the comments. I'd love to have these discussions with you. I really enjoy my discussions in the comments section that we do. So really feel I would love to see you guys make comments in there and keep this conversation going. All right, you guys have a great time, and God bless.
Drive